Hello friends, welcome to yet another intriguing edition of Biology Made Easy. Today I'll be bringing you a very ecstatic topic known as the regulation of the entire glycogen metabolism, comprising of the regulation of glycogenesis, which is the formation of glycogen, and the glycogenolysis, which is the breakdown of glycogen. In my previous two videos I have explained in depth the molecular details, the biochemical details, covering the glycogenesis and the glycogenolysis. So now if you are a novice in biochemistry, let me tell you that the regulation is the most important and most significant stuff that goes on. Without regulation, any process would keep on going and would keep on getting seized. So you don't want it to happen. You want an optimal balance. The biology is all about attaining the optimal balance in the body. We don't deal in extremities. We need to maintain, we need to acquire optimism. Whereas we need to maintain the optimal concentration or optimal balance between the concentration of the molecules every time. You can see any pathological reports where you will be looking at a specific range. It's not 0, it's not 500. It's like, just for example, the concentration of glucose must be from 70 to 110 mg per dl. It should not be 0, it should not be 500. Whereas you will suffer from either hyperglycemia or hypoglycemia. So now let me get down to business. Number one, we have already dealt with all these enzymes. Glycogen phosphorylase. It is needed to commence the process of glycogenolysis, which is the breakdown of glycogen. Now glycogen phosphorylase A is the active form of glycogen phosphorylase. In its active form, its serine residues which are present already in the glycogen phosphorylase are phosphorylated. When the serine residues are phosphorylated, present in glycogen phosphorylase A, then glycogen phosphorylase is known as glycogen phosphorylase A, and it is the catalytically active enzyme which will do the degradation of glycogen molecules. Then how does it get activated? First of all, we know that there are two enzymes. One is glucagon, two hormones. One is glucagon, another is the epinephrine, also known as the adrenaline. These two hormones bind with the G protein coupled receptors, GPCRs, present on the different cells of liver, muscle, kidney. So what happens is, when these hormones bind to the specific GPCR, it leads to the activation of adenylyl cyclase enzyme, which converts ATP to cyclic AMP, which is a secondary messenger. I have reiterated it many times in my previous videos. CAMP is a cyclic adenosine monophosphate, that is the secondary messenger. And CAMP in turn, via signaling cascade, activates a CAMP dependent protein kinase known as protein kinase A. Protein kinase A, as we know, there are many types of enzymes. There are 6 types of enzymes, whereas the kinases have the capability to induce phosphorylation, that is, transfer phosphate group from ATP, making it converted into ADP, and transfer that specific phosphate group to a specific compound. So it transfers phosphate group to the glycogen phosphorylase B at the serine residues, converting into the catalytically active glycogen phosphorylase A. Now how does it do that? What will happen is, first of all, pKa would directly not transfer it to glycogen phosphorylase A. What it would do, it would phosphorylate glycogen phosphorylase B kinase. And when it does phosphorylate glycogen phosphorylase B kinase, the glycogen phosphorylase B kinase gets activated. Now glycogen phosphorylase B kinase, after getting activated, since it's again a kinase molecule, it would then phosphorylate glycogen phosphorylase B and convert it into glycogen phosphorylase A by phosphorylating the serine residues and make it catalytically active. So that's how the regulation process goes on. One more thing. Why do we need to go for glycogenolysis? Because sometimes we have higher concentration of AMP, adenosine monophosphate. That means we have lower concentration of ATP. That's why we need to break down glycogen, get glucose upstreamed, and then glucose would be streaming down the pathway of glycolysis and Krebs cycle to increase the amount of ATP, decrease the amount of AMP. So if AMP concentration is high in the cell, that means we are going for glycogenolysis. AMP is an allosteric activator. AMP would activate glycogen phosphorylase A, whereas if ATP concentration is what is called high, that would down-regulate glycogen phosphorylase A, because if you have enough ATP, why would you like to go for glycogenolysis? So it's an allosteric down-regulator or allosteric inhibitor, you can say. Number 2, this process cannot go on forever. It has to be ceased. It is not incessant. So when ATP becomes, concentration of ATP becomes high, what happens then? The PP1, right? That is phosphoprotein phosphatase 1 becomes active. One more thing, if glucose concentration becomes very high, that is after the breakdown of glycogen, what we get is glucose residue. If glucose concentration becomes very high, glucose would again activate PP1. And PP1 would do what? It's a phosphoprotein phosphatase 1. It would degrade the phosphate group, since it's a phosphatase. It's a protein, it's an enzyme which would degrade the phosphate group and convert these phosphate groups on serine would be dissociated, and glycogen phosphorylase A would be returned to glycogen phosphorylase B, and therefore this process would be ceased. This is the main deal here. Number 2, when we talk about glycogenesis, the main enzyme which comes to my mind is glycogen synthase. Now glycogen synthase, again, the glycogen synthase A is the catalytically active molecule, active enzyme. Now glycogen synthase A differs from glycogen phosphorylase A in a way that glycogen phosphorylase A needs to be phosphorylated at serine residues. But glycogen synthase A, even though it's catalytically active, but it doesn't need to be phosphorylated at all. If it is not phosphorylated, then it's called glycogen synthase A. Next, if it is phosphorylated on the hydroxyl group of serine, then it would be termed as glycogen synthase B, which is catalytically inactive. Now this very enzyme glycogen synthase can be phosphorylated, can be induced the phosphorylation by 11 different protein kinases. So I will talk about 2 different protein kinases here. Number 1 is glycogen synthase kinase 3, that is GSK3. GSK3 can induce phosphorylation on the CH2 domain of glycogen synthase on the serine residues and convert glycogen synthase into glycogen synthase B, which is catalytically inactive form of glycogen synthase. But GSK3 on its own cannot cause this very reaction. It's dependent on another protein kinase known as casein kinase 3, which induces the phosphorylation of serine residues on the nearby serine, which is phosphorylated by GSK3 on the vicinity. So it would phosphorylate first. It is known as the priming reaction. It would prime for the phosphorylation to be induced by GSK3 glycogen synthase kinase 3. So first of all, the phosphorylation must be done by casein kinase 2, which would prime the reaction, and then glycogen synthase kinase 3 can then phosphorylate the glycogen synthase, which is glycogen synthase B, catalytically inactive, right? And the conversion would be done from glycogen synthase A to glycogen synthase B after phosphorylation. So then what would happen is this enzyme would get, this glycogen synthase would get inactivated glycogen synthase B. But if you want glycogenesis to take place, to go on, to start, then what you need is to convert glycogen synthase B from the phosphorylated state to the unphosphorylated catalytically active state known as glycogen synthase A form. How do you achieve that? We all know that insulin is the most important hormone which triggers glycogenesis, and glucagon triggers glycogenolysis. So once insulin binds to the IRS, which is a type of tyrosine kinase receptor, it would activate PKB, which would be dealt in detail in my later videos. The complete signaling cascade covering the intricate details of insulin and IRS signaling, insulin receptor signaling. So what happens here is the insulin molecule or the insulin hormone binds to the insulin receptor, which is a type of tyrosine kinase receptor, and causes the activation of PKB of protein kinase B. It's again a kinase which is extremely inclined or extremely conformationally inclined towards GSK3, glycogen synthase kinase 3, and it would cause the phosphorylation of GSK3 on the serine residues. If this phosphorylation is done, then GSK3 is rendered inactive. If it is rendered inactive, it cannot phosphorylate glycogen synthase, and then glycogen synthase in the unphosphorylated state would remain glycogen synthase A and glycogenesis would be turned on. Next thing, if it's allosteric activator, glucose 6-phosphate is also present, then also this process can take place. Another thing is, this phosphorylation tips the balance towards the dephosphorylation by PP1. Here dephosphorylation did what? It switched off glycogen phosphorylase A. But here dephosphorylation by phosphoprotein phosphatase 1 would switch on glycogen synthase, converted it into the active form known as glycogen synthase A. One more thing I would like to tell you. Glycogen phosphorylase A, when it is completely active, right, it inhibits PP1 because it is active, it needs to inhibit it. If the PP1 is inhibited, the PP1 cannot dephosphorylate glycogen synthase B, which is the inactive form, and convert it into the active form glycogen synthase A. Therefore, glycogen phosphorylase, in a way, what it does? It inhibits, it downregulates glycogen synthase, and the body really wants it. Just be prudent. If the glycogenolysis is upregulated, you don't want glycogenesis to be upregulated at the same time, and vice versa. One more thing I would like to tell you on the hindsight is, in specifically the muscle cells, for glycogenolysis to happen, the calmodulin plays a great role. In order for glycogen phosphorylase to be activated and called glycogen phosphorylase A, which is phosphorylated on its specific serine residues, in muscles the allosteric activator is the calcium ions also. The calcium channel, ryanodine, receptors and all, they induce the incoming calcium to come in through the calcium channel or calcium voltage gated channels. Now calcium channel has induced the incoming or influx of calcium ions. So calcium 2 ions bind with the calmodulin molecule, right? Binds with the delta subunit, also known as the calmodulin molecule, on the muscle. Calmodulin is a specific calcium binding protein. So once it binds, then this protein, this entire protein complex, acts as an allosteric activator of glycogen phosphorylase A, right? So it does activate glycogen phosphorylase A. And here on the hind side, if this glycogen synthase kinase is phosphorylated, what happens is, if you want to get down into the molecular details, the phosphorylation of glycogen synthase kinase 3 on the serine residues, what it does, it activates the loop. Then this will fold and bind to a specific portion of the glycogen synthase kinase itself, which would prevent it from binding to the phosphorylated site of casein kinase 3, because casein kinase 3 has phosphorylated already at the nearby residues, and when the glycogen synthase kinase has a specific site which interacts with the primed phosphorylated site, and then it gets activated, and then glycogen synthase kinase would start phosphorylating. But if it is already phosphorylated, it would form a loop, and then it cannot interact with the primed casein kinase 2 induced phosphorylated sites, right? So if it cannot do that, it would become completely inactivated, and it also cannot present the surface for further phosphorylation. So that's about it, the comprehension and the intellectual prowess needed to understand, to comprehend the glycogen phosphorylase A activation and glycogen synthase A activation. Now if you have understood and were able to comprehend this video properly, kindly hit the like button, and if you have any queries, then kindly do not hesitate to post your queries on the comment section below. And if you found this content relevant, kindly subscribe to my channel, Biology Made Easy. See you soon. Thank you.